Guys, let's get started. Guys, just quickly, a quick update. Actually, a lot of you are really doing great. It's actually really nice to grade exams where the vast majority of people are just putting check marks on the page. Exams went really well. So I have graded all through, grading from the back forward. Not only was it easier, I think, to do those questions, but they were easier to grade. I graded all through question 5 and part of the way through question 4. That's where we kind of hit the first speed bump a little bit on the shifting part of the sequencing question. That's okay, it's just 5 points. And a whole bunch of people got at least something for it. Anyway, I believe that I should have them back by Monday. That seems very realistic on this time frame, unless something dramatic happens. So I'll keep you updated about that. And then if I do, I will return them Monday after class, Wednesday after class, Monday after class, Wednesday after class, so it's 2 weeks. And then after that, it's, whatever. You want your exam to come when you get it from it, I guess. So, okay. Also, I have to post problems at 6. Homework has been posted. That one is the next week, but I just wanted to get that out of the way. So if you think we're running something kind of weird about the homework schedule, or is, I just got to post it. Anyway, let's pick up and finish our unit on DNA recombination so that we can move on to our next biomolecule, one that does more than DNA. What we were talking about when we left off before the exam was the idea we first addressed last semester in antibodies. Antibodies are these proteins classified as immunoglobulin proteins because they contain, or they are built up of multiple copies of these 2 structures you see here, called immunoglobulin domains. And their purpose, you recall, is to bind to pathogens or antigens, more specifically, that are present inside of our body, and then biology happens. We really did not address the issue of the biology of the antibodies because that's a bit beyond the scope of what it is that we're going to do. But one thing that we did know that was quite remarkable about antibodies is that they could bind very, very tightly to these antigens, particularly at the epitopes. So they could bind quite tightly, but also with tremendous specificity. So you have a very high specificity, and you have a very high affinity. And those 2 things can be related, although they don't have to be. And this really necessitated a very, very close molecular interaction between the antigen, specifically the epitope, and the paratope on the antibody, which is great, because from a biology perspective, that's exactly what you want, right? You want to bind tightly to some antigen so biology can happen. But what we also noted last semester, we didn't address at all, was the idea that if you look at all the antibodies you make, and all the antibodies you possibly can make, over the course of your lifetime, the amount of information required to make all those antibodies would exceed the amount of DNA that's actually present in your cells. That is, you can, and in many cases will, make a substantial portion of your total antibody possibilities, but you don't have enough DNA to do it. And that's significant, of course, because remember, it is differences primarily in the CDR1, CDR2, and CDR3 loops on both the heavy chain and the light chain that give rise to this incredible specificity and high affinity, which is really paradoxical. You can make them, you're here, you're alive, so you are making them, but you don't have enough DNA. And so the question is, how is it possible to make essentially more proteins, or more possibilities of proteins, than you have DNA for? That should exceed really the information content, at least of the protein coding component of your genome. And to give you some indication about this, we can look into the genome of a metazoan that has an antibody system that's like ours, and we can see something really very interesting. So when we talk later about how bacteria arrange their genes, we'll see that bacteria tend to arrange their genes in something called operons. That is, genes that all carry out some common purpose or arranged together on the bacterial chromosome. That's very efficient. But in general, you're not like this. If you have 3 or 4 different genes in a pathway that make proteins, I should say, that are part of the pathway, they could be on all different chromosomes all over your genome. So we don't have that efficient organizational system for most of our genes. The exception is antibody genes. Antibody genes, the genes that are necessary for making all those wonderful antibodies, really are clustered in a very, very few positions or places in your genome, and we call these antibody locuses. And what we notice, very interestingly, is that if we look at these locuses in the genome, the different pieces or parts of the antibodies that will come together to make the entire antibody, we also notice something really confusing, which is that rather entire antibodies, which is what you would expect if the genes were coded for in the genome, we have sequences for parts of the antibody and multiple versions of those sequences with some small slight differences between them. And so if you look here, for example, at the heavy chain, you could see that, remember we have a variable domain, a variable heavy one domain. You would expect there'd be a sequence, right, for that particular part of that antibody found in your genome. In fact, we have many of them, 40 in fact, as it's shown up here, very related sequences for the same part of the antibody. And in fact, if we go through and look at the entire antibody locus, we can actually map where these sequences are onto the positions of the final antibody. And note again that for pretty much every single part of an intact antibody, there are multiple sequences in your genome that can code for that, right? So instead of just having one particular, for example, set of sequences that could code for the CDR loop containing regions all the way in the light chain, we have many of them. And so this kind of starts to propose a model for how it's possible to make so many different antibodies, right? We have sequences that are similar but not the same for each of these different parts. And so you could start to think about this as kind of like being a buffet. I want to make an antibody. So I'll take one green, one orange or yellow, one red, connected together with one blue. And if you look at all the possibilities that are there for these different components or parts of the mature antibody, you would see that gives me a tremendous amount of variation, right? I just pull one of these, one of these, one of these, one of these, and somehow get rid of everything else from the genome. And that's how I'm going to make my antibody. And so you can see there are, for both the light chains and the heavy chains, multiple components, and each of those components correspond to a discrete part of the antibody, right? So we have multiple DNA segments, and you pull one of these out, one of these out, one of these out, one of these out, you connect those together, get rid of everything else, and now you would have the DNA to make an antibody. And in fact, it turns out that that is exactly, remarkably, how it's done through a process that's known as VDJ recombination. And the nice thing about this is that even though the steps may seem a little different or distinct from anything you've seen before, it actually, most of this are just borrowing concepts, like you could say, or borrowing mechanisms from other biological processes that we've looked at. So again, just to illustrate to you how this whole thing seems to be working, we could look at the actual enzymes in a bit. We have an antibody. We want to make, of course, a heavy chain that has those three constant domains, and a light chain, and a variable domain that will also have some diversity in it. There are individual parts of the genome. We're going to pull those pieces out of the genome, pieces of DNA somehow out of the genome, get rid of the rest of the antibody locus, that all has to be gone, and connect our pieces that we want together. And I will tell you, before we look at the mechanism of this pulling and selection process is completely random, right? So you could take the first one of the first part of the genome, the second one of the second part, the third one, or the fourth one, connect those together, and make a whole host of different antibodies. And so this buffet approach, or this ingredient approach, is really where we get this diversity. The question is, how do you know which parts to combine, right? So we're going to propose that somehow enzymes are going to get into your DNA and know I need to take one of the reds, one of the greens, one of the yellows, and one of the blues, connect them together, and throw everything else. Okay, that's what we're saying that there's an enzyme that has to be able to do. The question is, how does an enzyme know, because obviously it's not color-coded in your genome, how does the enzyme know to take one red, one green, one yellow, and one blue, and get rid of everything else? To see that, we need to focus in a little bit on the actual sequences that are present, okay? We need to take a look at the sequences that are actually present. And if we do this, what we will notice is that at the end of each one of these segments of sequences that are actually end up in the antibody, there are extra little pieces of sequences. These aren't going to make up the antibody, but these are called recognition signal sequences, and they come in two different flavors, a 12 and a 23 flavor, we call them. And what defines these 12 versus 23 flavors? Well, both RSSs, recognition signal sequences, of the 12 and 23 variety have a heptamer and a nonamer on opposite sides of one another, right? So you can see there's a heptameric sequence and a nonamer sequence that are the same on both of these sides. Then, Separating out these distinct sequences, there are 23 bases of, doesn't really matter because it's all going to be gone anyway, just a 23 spacer nucleotide. It could be all A's, ATs, ATGCs, it doesn't really matter, or a 12. Okay? So again, outside of the parts that are going to really end up in the antibody, you have these recognition signal sequences in the DNA genome. This is part of the genome now, and they either have 23 spacers or they have 12 spacers. And if you look carefully, you could start to imagine that you could somehow pair up a heptamer of a 23 with a heptamer of 12, right, by just swapping the DNAs, and a nonomer of the 23s and 12s, and that's going to sort of make the DNA fold or align with respect to each other. You can't literally just pair them up. You're going to have to turn some of them upside down, right? Anyway, this gives rise to what we call the 12-23 rule of antibody biochemistry. What is the 12 to 23 rule? The 12 to 23 rule is that you always pair a 23 with a 12. That is, you can only pair a VH, for example. You can see here one of the VHs, and remember there are many, right, VH sequences, which when you pull out of the genome is totally random. It can only be paired up with a DH because you have to combine a 23 with a 12. Can you combine it all the way over here, a 23 with a J? That's going to make the little piece all the way in the CDR loops. No, you can't, because you're not pairing a 23 with a 12. So when it was first noted how these things are being rearranged, it was noticed that in the genome, a 12 always paired with a 23, and that's what we call the 12 to 23 rule. And that's how the segments that are going to be connected become connected to each other. So that we don't, for example, have an antibody with 2 reds, no green, but 1 yellow segment, right? That can't happen because we wouldn't be pairing 12 to 23. Anyway, there are a whole bunch of different proteins that are involved in this process. Some of these we'll actually have seen before, some we've seen, we just haven't named them. You should make sure to review this, mostly to understand the mechanism, not so much just to know the naming of them. What, again, we're going to do is we're going to see how now we go and take these segments and actually connect them, so that's what the complex is. So we start, for example, with a part of the genome that's going to look like the top, and then we end with a part of your antibody genome locus that's going to look like the bottom. So in other words, in the process of doing this, something dramatic is going to happen to the genome of the cell in which it happens, which is that it's actually going to get shorter. The genome is actually going to get smaller because we're going to throw out a lot of extra DNA in the process of a B cell starting to actually make an antibody. The other thing, which is related to that first thing, is that means that if you were to sequence the genome of one of your B cells, it would have a different genome than another one of your B cells. You don't, in fact, have a single genome, particularly in your B cells, because different pieces of DNA have been thrown out and others kept and connected to make the mature antibody. Anyway, here's the rule, again, to show you the 12 to 23 pairing that we're going to establish, right? You can see how we can imagine pairing heptamers from a 23 with a 12, as well as the nonomers, but we can't do so directly. We can't just pull a heptamer and a 23 and a nonomer and pair that with a heptamer of a 12. There's got to be some twisting of the DNA taking place. The antibody that's going to do this is one that you do need to know, is known as the RAG complex, the recombination antigen generation complex. This one you do need to know, and you can see here kind of exactly what's happening. Notice that we've taken one B segment out, and we've taken one J segment out. Those pieces are going to end up in the antibody, right? That's going to represent one part of the antibody, the B, the J is other, and everything else is going to be tossed away. Notice that the enzyme complex, its first major job is to pair the nonomer of the V with the nonomer of the J, and the heptamer of the V with the heptamer of the J. But to do that, it has to actually fit onto the enzyme, right? So this requires a specific shape. The enzyme will only allow the two 12s to come together and the two 23 pieces to come together, those spacings. This has to fit on the enzyme. And so when the 12s and the 23s come together, this is how the V and the J that are going to be combined are now positioned next to each other. This is a pretty good representation, but there is one thing that it kind of misses. And you see like the green bit at the top, the nonomer, it looks like there's just a lot of DNA there. that can actually be kilobases of DNA, okay? Because you're going to see we are going to be throwing out everything between that V and that J. So even though it looks like there's just a little bit of DNA that's fitting on there, there's actually a huge amount of DNA that we're going to actually get rid of. Okay, so how is this going to work? The first thing that the Rad12 complex proteins is going to do is to pull out one V and one J. Just going to randomly pull them out of the genome. There's no order to this, there's no anything to that. It's going to bind to the DNA and just loop these two segments out. And when it does so, it's going to bind, as you see here. We're going to pair the heptamer of the V with the J, and we're going to pair the notamer of the V and the J, and the correct orientation is going to be established by the fact that that's the only way to get the 12 and the 23 spacers made. Right? So essentially, the purpose of this enzyme is to use those base pairings between heptamer, heptamer, notamer, notamer, as well as the positions of the 12 and 23 segments to fit onto the enzyme, such that the V and the J are now paired next to each other. Now the enzyme is going to actually start to do something enzymish, because enzymes have to do things, not just bind to their substrates. And so it's going to do two activities. It's actually going to cut, right? Notice that it cuts out, right, before the heptamer, right, on the V and the J. It actually cuts those sequences, and then it's going to seal the ends together to make really a single strand of DNA. So notice that we have what are called hairpin loops now. The 5' of the V has been connected to the 5' of the 3' of the V, the 5' of the J connected to the 3' of the J. All of the intervening DNA, which looks like just a little bit of DNA, but is really a lot of DNA, right? All the other V's, all the other J's until we got to that V and J, they're gone. They're out of here. They're completely lost. We're never going to see them again. Notice that we have these hairpin loops. Really critical now to point out, although the slide that this is the part that it doesn't characterize very well, when it makes this cut, it doesn't do the world's best job. It's not that super accurate, the cutting part of the enzyme. So it looks like it's making a nice, clean, beautiful cut. It's not. It might cut here, it might cut here. So the result is that the sequence now in the V and the J might have lost a little bit of DNA that was originally there. Okay, next thing that has to happen is to cut those hairpin loops open. Notice that we're going to cut those hairpin loops open to reestablish them so that we can join the V and the J. We can't join them right now. They are hairpin loops. Remember how I told you the cutting of this is less like a fine knife than just kind of hacking at the DNA with a chainsaw maybe or an axe? It's going to do that again. So instead of making nice, beautiful, clean cuts, leaving two beautiful blunt ends that we can somehow have together, or maybe having some homology between these if it's there, it's just going to whack at it. So that's exactly what it does. It chops off. But notice that in the chopping process, it leaves some DNA damage. This is not a clean, efficient system. This is just whack those hairpin loops open. And some DNA could very well get lost. Now, what we want to do, of course, our purpose was to join the V and the J together, because the same thing would be happening with the J and the next segment over. We're just looking at one part of it. But what do we have here? We have here some DNA damage. Fortunately, we've already seen how to repair DNA, right? We can't just join the V and the J together. We need to actually fix them. We need to bring them together. And so that's kind of what's actually going to be done here. And so here's the idea. Once we generated the hairpin loops, that's the second panel down, we cut them open. Notice that the cutting was pretty rough. We can't just join these together because there's really no homology between one side or another. And now things are going to get really sloppy. And this sloppiness, in fact, is going to be why we can have so many different antibodies in your genome. Because the way they're going to be repaired is by a version of NHEJ. Now remember, I told you when you're fixing your genome, NHEJ is just a last resort. NHEJ is generally just going to work by bringing the two halves of DNA together, and if they don't quite have some homologies, an AT on this side with a TA on that side, it's going to chop pieces off and then try to bring them together again until you get a couple of base pairs overlap between the two parts of the DNA that you want to seal back together, and then it's going to be ligated. So this is going to use an NHEJ-like approach, but it's also going to do something else. Because NHEJ would already introduce changes into the sequence, but to just make sure that you have lots and lots and lots of diversity actually happening, another protein which is not involved in regular NHEJ, but which is in your immune cells called TDT is going to come in, and it is a very unusual polymerase. Remember, every other DNA polymerase that we've seen can only add bases, right, that match across either side of the genome using the template. Not TDT. TDT is just going to start throwing nucleotides at both sides. Throw it, throw it, throw it. Can we connect? No. Chop a few off. Throw a few more on. Eventually, by all of this really rough hacking and throwing nucleotides at either end, you'll get a couple of sequence overlaps, and then NHEJ can finish the process of sewing this in the middle. In the meanwhile, what has been done here with all of this chopping and then throwing random nucleotides is creating sequence diversity far greater than was ever originally present in your genome. So far, we've seen two origins of the ability to make more different antibodies than the DNA you have. First, we chose random segments. I've only showed you here now how only two of them are connected, but really all of these different segments would be connected by 1223 rules, right? So pick a random one out, connect them together, but in the process of generating the joints that are going to be formed, cut them just very roughly so you can create essentially initially losses of DNA, but in random places means you're creating random DNA sequences, and then when you're bringing them back together, use the least effective DNA repair mechanism, and just to create more sequence diversity there, use the one DNA polymerase that just throws nucleotides at it until some sort of homology is actually found. And so that's the second approach, right? The first approach is the random pulling of segments. The second approach is the process of cutting, resealing, but doing so very error-prone. And even that would not be enough DNA, actually. Even with all of this, you still couldn't make enough antibodies. So there has to be one other thing, at least, that accounts for all this antibody diversity, and there is, and this is a process that's known as somatic hypermutation. So the idea of somatic hypermutation takes advantage of something you actually saw on your last slide. Notice that these Cs are very easy to deaminate. They'll happen spontaneously in water anyway, but in your white blood cells, or particularly the B cells, you actually have an enzyme that speeds this process up. So it binds actually to the DNA by mechanisms that aren't completely understood, close to where the DNA would be for the CDR loops, because that's of course where you want most of your diversity. You don't really need very much diversity in other places. And it catalyzes the deamination. And so what can possibly happen after a deamination? Well, there's a couple of possible events now that you've gone to U. One possibility is it replicates. No repair happens. It just took what was supposed to be a C, so pairing with a G. Replication happens. We know these white blood cells are dividing quite rapidly. DNA repair never occurred. And so you ended up with a little bit of sequence diversity there. Another possibility, of course, is that you can get base excision repair. Sometimes it actually gets fixed. But the base excision repair in your white blood cells, particularly I should say the B cells to be precise, tends to be a little bit less precise than others. Nature has selected it to be such. And so the base repair can happen, but it doesn't always repair it quite the right way. It takes the wrong base out, but doesn't always quite put the right base in. And because these cells are dividing so rapidly compared to most other cells in your body, oh well, the fix never happens. But then even that is not enough. It turns out that you also have in your B cells an error-prone polymerase. So sometimes you try to do a true repair, remove whole portions of the DNA, right? You try to remove portions of the DNA, and a very error-prone polymerase comes in, one that just makes lots of mistakes, and it actually can change a whole bunch of nucleotides. So these are the mechanisms that account for the fact that you have more DNA, less DNA, than the proteins that you end up making. One, the buffet approach, right? Take one of these, one of these, one of these, one of these. You have lots of different segments corresponding to each diverse part of the antibody. That allows you to have multiple options. Two, the process by which those segments are assembled is sloppy. It is possible to lose quite a substantial amount of DNA, and in fact, many of the antibody-generating sequences never end up making antibodies because the DNA that they code for does not code for viable proteins. This is very inefficient, and this is lost. But if an antibody does make it to this point, the last opportunity to diversify that sequence is through something called somatic hypermutation, where the C's get intentionally deaminated to use by an actual enzyme that does this, and then the DNA repair has trouble keeping up with the speed in which the cells are dividing. The consequences of this is that quite a number of errors actually end up in the DNA, and this is where you get the diversity of your antibody sequences. This is how you get more proteins than the DNA actually have for them. Anyway, okay, good. So that is the end of our discussion of DNA for a while. Let us start with our next molecule, RNA. Very important, RNA, because we've seen that if we look in terms of your genome, even though only a relatively small portion of that actually gets coded into protein, a fairly substantial portion, 25, 26%, maybe a little bit more, is actually going to get the information, I should say, used to make a particular RNA molecule. So RNA is quite significant. One of the things, of course, is that some of that RNA will be used to code for protein, but as we'll see, most of that RNA actually has other functions inside of your cell. Before we go on, we do need to discuss the letter of the day today, which is S. Today the letter of the day is S. Why is the letter of the day S? In this lecture, maybe just a little bit, but not so much, but maybe a little bit. But certainly, as we look forward, we're going to be seeing a little bit of experimental data, or at least looking at the names of proteins and RNAs and so on, molecules that were discovered from doing actual experiments. And a lot of those proteins and protein complexes will be named with an S fixed after them. 25S, 13S, 16S. And so it's really critical that we understand what that S is actually meant to represent, because it has an actual functional significance. It turns out that that S is, roughly speaking, meant to measure the amount of mass, or the size of things, because we assume, not always correctly by the way, that since all biological molecules are made of carbon, hydrogen, nitrogen, oxygen, some phosphorus sometimes, and maybe a little bit of sulfur, that the size of a molecule is roughly proportional to its mass. And so this S actually refers to the term Svedberg, who is a great biochemist, and it's a measure of how much or how far down in a centrifuge tube a molecule or molecular complex will travel, right, if it's being centrifuged through a medium of a specific density, right? So if we take, for example, a whole cell, which is a mixture of many molecules and molecular complexes, we put it in a centrifuge tube, we include in that centrifuge tube a material that we know the density and the viscosity of. If we spin it really fast, the denser material will settle to the bottom, the less denser material will travel less distances, and so this is one way in which we can get a separation. And if we fix the amount of time in which we do that, the distance in which it travels will be proportional to the density, and we use that as a term of mass. And so that is where we see the S term. And why am I making such a big deal of it? Okay, whatever, 45S, 12S, 13S. Well, in normal math, the math that we're most accustomed to, 10 plus 15 equals 25. I mean, that would be a really easy problem to put on the test, right? But one of the things that we will see when we look at some of these molecular complexes is something like 10 plus 15 equals 17, which is not quite math, it should seem, right? If you take something that's 10S plus something that's 12S, you would expect it to be 22S, right? If we're saying that this is all about mass, one thing that has this much mass, another that has this much mass, bring them together, it should have the sum of their masses. And that's not what we're going to see at all. Because again, the density as it is moving through a centrifuge tube, mass is one of the determinants, but it's not the only one. The shape matters as well. So for example, if you have something that is very, very dense but compact, it could move faster than something that's less dense but more spread out. And so the result of this is that when we are combining together these different complexes of proteins and nucleic acids with S values, it's not always going to be the case that those S values add, because now not only has the mass or the density of them changed, but so has the shape. And so this is why we're talking a little bit about S now, because we'll see a little bit in this lecture, but more moving forward. Complexes named by S. We are using that S as kind of a surrogate for the mass. Something that has a higher value of S is heavier, lower value of S is lower, right, less massive. But just keep in mind, it's It's actually the mass, but also the shape, because of course something that's very, very spread out is going to move much more slowly than something that's nice and compact. Okay, just to remind us a little bit now about the RNA molecule that we're dealing with, here are the bases. Again, you've seen all these, and they appear on the fact sheet anyway, and hopefully now you all know why it's U that appears in RNA, but T that appears in DNA, so that's something that we've covered extensively, so at least on the test, so we don't have to discuss too much more. Not going to talk individually about what each one of these different types of RNA do now, but I do want to just illustrate that other than just the messenger RNA and the tRNA that, of course, we've encountered a lot in biology classes, and maybe before that, there are a lot of different types of RNA, and they play a role in so many different processes. Probably the most remarkable one is that the actual enzyme, the ribosome that we'll talk about next, although it is a complex of proteins and RNA, it's actually the RNA that does the catalysis to make the peptide bonds. RNAs also, we'll see, play an important role in regulating the expression of other RNAs, that is, whether which RNAs do or do not get made as a result of their being in the genome. So yeah, I mean, these are essentially, lots, and probably not even conclusive, of all the different types of RNAs. And it's significant because even though these are all RNA and they all have ribonucleotides, there are functional specialities here, and we'll see that not so much in prokaryotic cells, but certainly in eukaryotic cells, having different types of RNAs means having to have different types of regulation, which means there's not a one-size-fits-all factory for making those RNA molecules. Anyway, just one kind of fun RNA to look at is this one. We don't know what it does, to be honest, so I'm not showing it to you because it's an RNA that has a really cool function. But it is very interesting RNA. It kind of illustrates just how much more there is to learn about RNA biology and how important it is. This is the HR1A, R1A RNA. It's found in the nervous system and the brain, actually, I should say, confined to the brain during development. And so that's probably really significant. But what's really significant about it is that this is an RNA that has changed dramatically, not so dramatically, I should say, from every other organism up to chimpanzees. And then from chimpanzees/bonobos, which is our nearest closest relatives, up to humans, all of those different nucleotides that you see here have suddenly changed. So there is this huge gap, in other words, in an RNA that is selectively expressed in our brains that go back to our nearest cousins that we are related to evolutionarily. And so, why that is, I mean, I don't know. Certainly this is not implying that this is the only RNA that makes us distinct or have all these remarkable capacities in our brains. But the fact that we do know that it has these changes, and that these changes do correspond to remarkable evolutionary changes in human brain structure, or the structure, I should say, of primate brains, is probably interesting and important. And so, just illustrates to us that even though we can know a lot about the chemistry, I think, of these RNA molecules and even the shapes they take, there's still a lot more to be learned. Anyway, here's one that we do know a bit about. Has anyone ever heard it exist, by the way? This is something you guys covered or talked about. Did anyone ever hear about X silencing? That's something you talked about in biology, the idea of X silencing? Okay, great. Got work to do. Here we go. Here's the thing. Most of your chromosomes are the same, right? Biological males versus biological females. But we know biological females have XX, biological males are generally XY, and that creates a problem because there are genes on the X chromosome. And it is really, really important that whether you are a biological female or a biological male, many of those genes that are expressed not just on those chromosomes, but in general, be expressed at a certain appropriate level, just the same amount. In other words, too much or too little, both of those situations are really bad for you. And so this would create a problem for biological females, right? And biological females would effectively have twice as many genes on the X chromosome active as biological males do, and they would therefore have twice as much gene activity, and that can be quite problematic. It's not always necessary that to the case that more gene activity is a good thing. Sometimes more gene activity can be a bad thing. In fact, we know that because when this system does not work properly, it actually leads to things like breast cancer and ovarian cancer. At least it's correlated with those things. Anyway, so here's the thing. Biological females have two X chromosomes, but we only want to express genes from one of them, and the second one should undergo what we call X silencing. In other words, the chromosome is still there, but genes are no longer expressed from it. In fact, it becomes something that's known as a barcode, no longer capable of being read and having RNAs read off of it. And the question is, how does that happen? And it turns out that there's a bit of competition between the two X chromosomes. One of them wins, obviously. It's the one that's going to have its genes expressed. But it's going to actually make this RNA called exist. And what that exist is going to do is coat the other X chromosome, and essentially that signal of having all that RNA coating that other X chromosome is going to bring in a complex of other proteins and potentially other RNA molecules as well, and essentially crush it to death. That's just the process of X factor silencing, right? Two X chromosomes, one of them, which everyone, I guess, starts off first, will make this exist RNA that will coat the other chromosome and essentially just kill it, take it out of remission so that genes can no longer be expressed from it. And now you have the same amount of gene expression off of the X chromosomes in males, biological males, apologies, as you do in biological females. And so this is called X silencing. It's another really interesting thing that RNA does. And again, it is RNA molecules that are actually doing this coding, triggering the process of the destruction of the second chromosome. Okay, other interesting questions we could ask are, we know that a substantial amount of the genome, roughly 25-26% in you or cows, I think we're precisely measured, code for RNA, but how much? Like, how much RNA do you actually have? That depends on how you want to count. If you look at RNA by mass, you can see here ribosomal RNAs, that's rRNAs, those are the ones that are the ribosome, the protein-making factory. Those are by mass most of the RNA. You look, however, by the total number of molecules that you have in any rough given cell. That would be tRNA with ribosomal RNAs being a relatively small proportion of that, detectable but relatively small portion. This obviously means that ribosomal RNAs must be huge, right? You have relatively few of them, but they make up most of your RNA by mass, so they are in fact gigantic. tRNAs by comparison are the most plentiful, but also relatively small compared to other RNAs that are in your body. Anyway, all of this gets multiplied by having about roughly 10 to the 7 total RNA molecules present per cell. So there's a lot of RNA there. Most of it, again, by mass is ribosomal. Most of it is by numbers is tRNA. Big and heavy RNA, not many of them, but big and heavy versus lots and lots of some of the smallest RNAs, which are tRNA. In terms of structurally, just to go back again, we've already talked about the differences between having T and U, so that's already covered. The other thing that's of note is that most of the time in RNA, we do not have our B form DNA. We actually have an A form. When RNA forms helices, they normally A form. The difference, you recall when we look back at some of those parameters, is that the A form is much, much more compact. The two sides of the grooves are roughly even in size as opposed to having one big, deep, major groove and kind of a shallow, narrow, minor groove in B form. The A form has grooves that are roughly symmetric on both sides. This happens because of the presence of the hydroxyl group on the 2'. Notice in DNA where we don't have that hydroxyl group, that allows the 2' to kind of shift into that endo conformation. Nothing's blocking it, it gets out of the way of the phosphate group and everything else. But if you have the hydroxyl group there, it's just going to collide with everything in the phosphodiester backbone and the remaining sugar. So that forces it into the C3 endo conformations, pushing that up and therefore the 2' down. This brings the two phosphate groups much closer together, the result of which is, again, we have roughly evenly sized grooves, but a much smaller molecule per number of base pairs. When we looked at DNA, we didn't see much of a change, right? Mostly we just had those 4 bases. There's a methyl group here and there, and sometimes one getting added by the diamethylase. This is not true when we're looking at RNA molecules. There are some RNA molecules that are just made of those 4 bases, but most functional RNAs actually turn out to have some sort of modifications. You do need to, we don't care about that. Do not need to know what these specific modifications are, with the exceptions of ones I'll specifically show you when they appear with us. You just need to know that it's really not correct to say that RNA is mostly made of A, G, C, and U, right? Most of those bases have been chemically transformed by other enzymes. Some of those enzymes are in fact RNAs. And this is both exciting and challenging. It's exciting because what it means, of course, is that RNA, unlike DNA, which is mostly confined to its 4 bases, all of these different functional groups mean that RNA can do all sorts of interesting chemistry, fold into all sorts of interesting shapes based on the types of interactions that we talked about last semester. But it's maddening because, of course, first of all, making these in the lab and introducing them to an RNA can be very challenging. And sometimes we don't even know they're there, right? I mean, they are discovered subsequently. And so they have important biological consequences. If you make an RNA in the lab, which you can actually do, you just use the 4 bases, it quite often doesn't maintain the same shape or function as the native RNA, and that's probably because these modifications are not present. They need to be put in, and that's actually experimentally challenging. We do mention the fact that RNA, unlike DNA, can take on a wide range of shapes. Here are most of them. What we call the double helix is just really a stem. That's when you have perfect lots of good base pairing. You can also have unpaired sequences that are present between those stems, and that's a hairpin loop or more complex structures in which two stems come together sharing a single strand. Sometimes most of the strands are base paired, but one is not, and that nucleotide actually flips itself out. That's a bulge. Two unpaired actually can give you another loop, and anyway, you can read these. These are just the secondary structures that are most commonly found in RNA, and yes, you probably should know these and how to recognize them. But just to be clear, just in the same way secondary structure does not capture the full structure of an RNA molecule or a protein molecule, same thing as an RNA. And I mean, I guess we've all seen both the cloverleaf structure of our tRNA, but also the attached structure. But now you can hopefully begin to appreciate what I mean. I mean, DNA, A form, B form, underwound, a little bit of overwound, or even supercoiled, is still pretty structurally homogeneous. This is not a molecule, right, that looks like anything structurally homogeneous. There are multiple loops, multiple stems here. That's really critical. You'll also notice, you don't need to know the specific ones and what the positions they're in, but notice how many of these modified bases are actually present in the tRNA molecule. And again, that makes studying these functional groups rather complicated, right? Because you can't just put these in using easy chemistry. So remarkably, there's still a lot we don't know about the tRNA molecule, including why it folds up into that particular shape. But it does illustrate the point. Anyway, so let's talk about how we start to make some of these molecules. If we do so, we're going to need a couple terms. I'm not saying there's not going to be a terms question on the test, but the purpose of these terms is really mostly just so that we can have a discussion, and I don't have to keep defining them each time. So let's just go through them. Not surprisingly, RNA polymerase, that is an enzyme that makes RNA, just like we saw DNA polymerase, right? So that's pretty straightforward. The most significant differences, we'll see some later, but here we're going to be using ribonucleotide triphosphates, not deoxyribonucleotide triphosphates. They will not accept deoxyribonucleotide triphosphates as substrates, so there is some selectivity there. The process of enzymatically making RNA is called transcription. There are laboratory methods for making RNA as well, using chemical synthesis, but we're not discussing those. So this is specifically using either in vitro or in vivo enzyme to do that. The transcriptome, that's all the RNAs in something defined. So what do I mean all the RNAs in something defined? Well, you have a transcriptome. All the RNAs that are present in you right now in all of your cells are your transcriptome. A couple things about that transcriptome. It's probably slightly different than the transcriptome when we started this conversation. So the transcriptome isn't necessarily a static entity. It in fact can be quite dramatic. It could change depending on temperature, time of the day, all sorts of things, right? You also have other transcriptomes. You will have a heart transcriptome, a liver transcriptome, and those two things won't necessarily be identical. And the reason I emphasize this so much is that controlling that transcriptome, that dynamic transcriptome, is of course an important regulatory feature in keeping you healthy and alive. So when this transcriptome regulation system that we're looking at goes off, bad things can happen. The RNA polymerase is going to need some place to bind on the DNA. It's not just going to set itself down anywhere and start the process of making an RNA. That sequence is called the protein, the promoter, sorry, where the proteins are going to bind. There are also operator sequences. These are sequences in which proteins that are going to prevent this process from actually happening are going to work. So that's the operator sequences. And we also have positive control elements. You could kind of think of them as almost unlike the opposites of the operator. These are the binding sites on the DNA now, where proteins that are going to make more transcription happen. So we have operators, which are inhibiting proteins that are